Yeah, okay. Good evening, Pankaj. Good evening. Good evening, Padmavati. Good evening, Saima. Okay, so friends, if every one of you quickly recall, in the yesterday class, we were discussing in the yesterday class and day before yesterday class, we were discussing about this particular cancer, and in just 10 minutes, we are about to finish this and we will solve some questions. Right. So we understand, friends, in the yesterday class especially, what we understand in the yesterday class especially, friends. Good evening, Savita. In the yesterday class especially, we understand the causes of cancer. We read radiation, we read chemical carcinogens, we read the biological agents. I hope every one of you recall, friends. Later we studied majorly about the mutations that cause cancer. Now we understand proto-oncogene also, as well as tumor suppressor gene also. I am just reminding to all of you, friends. Good evening, Kavita. When you heard the name of a proto-oncogene, the common point has to, good evening, Srikant. The common point has to keep it in your mind is dominant. When you heard the name of a tumor suppressor gene, what is the common point has to come in your mind? That is the recessive. Okay, later we understand everything. I strongly believe we have stopped exactly at this particular slide. Hello, Aishwarya. That is what, friends. Hello, Shrikant. Then we started at we stopped at this particular slide. That is the P53. If you all very quickly recall my words, what did I tell to all of you? P53 is not going to be released in the body every single time. P53 is going to be released in the body whenever there is a requirement. Otherwise, it is not at all going to be released. What are those conditions? You only try to understand. Under oxidative stress, impairment of hormones, several physiological responses got impaired. There is a nutrient deficiency, there is a low oxygen content, there is a damage of the DNA. Only during this particular conditions only, friends, we are going to use this particular P50, only during these conditions P53 is going to get activated. I hope every one of you remembered the moment P53 get activated, what it is going to do, friends? It is going to arrest the cell cycle, first step. After arresting the cell cycle, it will try to repair the DNA. Friends, if it is unable to activate the DNA repair mechanism, or if the repair was not done successfully, then immediately P53 allow the cell to undergo the apoptosis, then followed by the necrosis, that is cell death and tissue death. Friends, keep it in your mind about this particular PRB. We have discussed it already. What did I tell you, friends, about the PRB? Everyone keep it in your mind without fail. Protein retinoblastoma, which is the first identified tumor suppressor gene. I'll tell you in a very simple way, friends. I want every one of you pay attention towards what exactly I am saying right now. We all know very well, we use it to say that S phase is the phase where the DNA is going to undergo the process of replication or duplication. We know it. So what is the point I want you people to remember here? This particular S phase, listen to me carefully. If the DNA wants to undergo the replication in the S phase, first of all, it has to prepare itself at the G1 phase. Please listen my statements very, very, very carefully. What, friends? The cell has to prepare itself at the G1 phase. We all know very well, friends, if the cell wants to be prepared at the G1 phase, it is going to synthesize a lot of proteins. Because, my friends, many of the proteins are extremely required for this particular for this particular DNA replication. So, friends, how all these proteins are going to be synthesized? This is important point. These all proteins are going to be synthesized by E2F, which is a transcription factor. So, friends, everyone listen my statements carefully now. Suppose example, there is a no damage at all. There is a no damage. Then whenever there is a no damage, what generally happen, friends? This E2F transcription factor never be activated. Now E2F transcription factor is inactive. Then what is then E2F will be active. I'm extremely sorry. When there is a no damage, E2F will be active, and E2F is involved in the production of protein synthesis. By that the cell enter into the S phase. There is no problem at all. But now you people listen my statements carefully. Now what is the condition? Friends, the condition is DNA damage. there is a damage of the DNA occur, do you think we have to synthesize the proteins which are required for the S phase? No, because we should not allow the cell to enter into the S phase first of all. So what we have to do, we have to stop the preparation completely. So what should we stop actually? We have to block this particular transcription factor. What do we call it as, friends? E2F. Who is going to block this? Of course, PRB is going to block this particular E2F. Friends, whenever the PRB block the E2F, E2F never ever allow the synthesis of any kind of a protein. Hence, the cell never enter from G1 to S phase. Then we can protect the cell from the cancer. But here comes a very unique point. Just now I told to all of you, if the PRB wants to block the E2F, everyone listen my statements. The PRB should be in the unphosphorylated form every time. It should be in a which form? Unphosphorylated form. When the PRB wants to block the E2F. Suppose example, we should not allow the PRB. We don't want E2F to be blocked. Then you know friends, the PRB get phosphorylated. I want you to understand that friends, PRB will be inactive. When the PRB will be inactive, whenever it is associated with a phosphate group. In other hands, when the PRB is going to get activated, PRB is going to get activated when it is in the unphosphorylated form only. So please kindly do not forget that particular statement at all. So see here, this is the PRB. When the PRB is going to block the E2F, you only can see that, my friends. PRB is going to block the E2F when the PRB is what? Unphosphorylated form. Then only it can block it. Now you people only can kindly look into this. The PRB get phosphorylated completely. When the PRB get phosphorylated, what is going to happen? The E2F get activated. Two conditions you have to remember. When the E2F get activated, it synthesize the proteins required for the S phase. There is no problem. But everyone has to listen my statement. Suppose, friends, please correlate. Suppose there is a cell damage occur. DNA damage occur. There is a DNA damage occur. After DNA damage, once again the PRB in which form? PRB in the phosphorylated form. So what happens? Here DNA damage occur and PRB is inactive. When the PRB is inactive plus the DNA got damaged, then what is going to happen? This particular E2F transcription factor can undergo, can involved in the synthesis of many proteins, which leads to what, friends? Formation of a tumors. So you people always keep it in your mind. If we want to block the E2F, PRB all the time should be in the phosphorylated form. If the PRB is in the non-phosphorylated or unphosphorylated form, it cannot show the E2F. Then E2F can synthesize many proteins unnecessarily when there is no requirement, which leads to the different type of cancers. So you people normally remember protein retinoblastoma is exclusively involved in the blocking of a transcription factor named as what, friends? E2F. What is the function of a E2F? E2F is exclusively involved in translation or transcription and translation of many genes which are involved in the S phase. Very simple point. Next one, BRCA1, BRCA2. They are also very important, friends. Breast cancer gene 1, breast cancer gene 2. What are these two genes? These two are the specialized type of genes. Now, friends, I want you people remember one common point about the tumor suppressor gene. What is that common point? Every tumor suppressor gene is going to be activated in the cell whenever there is a particular adverse condition, or an example, whenever there is a particular damage. Same way with the BRCA1 and BRCA2 also. It is not going to be released every single time. So keep it in your mind. BRCA1 and BRCA2 are going to be released whenever there is a damage of the DNA. Please keep it in your mind, friends. Everyone, so how many number of copies are present that we have to remember without fail? Everyone has 2 copies of each of these genes. 1 copy inherited from each parent. So you only try to understand there is a mutation in these 2 genes. then what it will do, because of the mutation of these two genes which will involved in the development of a female and male breast cancer. But surprisingly, when compared to the male, in case of a female, it will cause more number of cancer. Not majorly is breast cancer, but the BRCA1 and BRCA2 associated with ovarian cancer, especially fallopian tube cancers, very dangerous cancers actually, even prostate cancers also. But when compared to the prostate, pancreatic and melanoma, this BRCA1 and CA2 majorly associated with the breast cancer, mostly in the female when compared to the male. Okay, now listen to me very carefully, friends. See here, initially there is a no damage. When there is a no damage, obviously BRCA1 and CA2 are not going to be activated. Simple point. Now you only try to understand, there is a damage occur to the DNA. Everyone look into this. There is a damage occurred in the DNA. Because the moment damage occurred in the DNA, friends, immediately BRCA1 and BRCA2 going to get activated. Now what the BRCA1 and CA2 will come? They will come and they will stimulate many kind of a DNA repair mechanism. Now what the DNA repair mechanisms will do? They will try to repair that particular DNA damage. Now look into this. So then there will be a no tumor formation. Now you people look into the other hand. Everyone of you look into the other hand. So BRCA1 and BRCA2 are got mutated. Now listen, there is a damage occur, but unfortunately BRCA1 and BRCA2 got completely mutated, friends. As a result, what is happening? It leads to a failed repair. Because of failed repair, immediately what happened? This BRCA2 immediately stimulated the P53. P53 stimulate P21, MDM2 protein, BAX protein. These are all what, friends? These are all type of tumor suppressor gene only. What I mean to say here, I mean to say when one tumor suppressor gene get inactivated, there is an activation of the another tumor suppressor gene. Do not forget that particular statement, which leads to cell cycle arrest. Sometimes it will take time. What it will take time? Everyone listen my statement. Suppose example, there is a DNA damage occurred. First, who recognized? BRCA1 and BRCA2 recognized. But what is the problem? These two got mutated. Now, before activation of P53 only, immediately what is going to happen, friends? This damaged DNA only undergo the replication. Then it leads to mutation. It becomes permanent and causes dangerous breast cancers. But you people has to remember, in our body what generally happen is, when one tumor suppressor gene got failed, immediately it is going to activate the another tumor suppressor genes. Please kindly do not forget that particular statement at all. Okay, so friends, those are all what? Different type of tumor suppressor genes and proto-oncogenes. The treatment and drugs, I want you to remember whatever the drugs I am marking actually. You all know, friends, nowadays we are having advanced type of cancer treatment. We have general treatments like surgery, radiation, chemotherapy. These three are most common. Now we are having immunotherapy, hormone therapy, the most accurate ones, targeted therapy and stem cell therapy. When compared to the stem cell therapy, targeted therapy will work very effectively, because in the targeted therapy, friends, we are going to exactly focus on the area where the tumor got formed. See, you have to understand, my friends, do you think this particular targeted therapy will be effective in the third stage and fourth stage, which are advanced type of stages? No, my friends, it's not possible. What I want you people to remember here is that the third and fourth stages, targeted therapy cannot occur. Why? What is the reason? Because I already told you, third and fourth stages, it's already undergone the metastasis. Correct, na? It already moved to various kinds of organelles also, surrounding organelles. Then how many targeted therapies we can perform then? That cannot be effective. So this targeted therapy will be effective only in the primary stages actually. Friends, because we know, in our exam, Even in the GATE exam, JAM exam, we get a direct question from the drugs. Okay, hence I am requesting all of you just remember the name of the drugs. Whatever I am marking, I want you people to remember those only. Okay, now first anti-metabolites, whichever I am marking, you people remember those: 5-fluorouracil, 6-mercaptopurine, methotrexate. I want you people to remember these three. Antibiotics, you remember only one specialized antibiotic, doxorubicin. Alkylating agents, please remember friends, alkylating agents are actually very important. The powerful alkylating agent which was asked in the exam also, that is the chlorambucil, phosphonamide and streptozocin. Please remember these three. No friends, remember mechloroethamine also. Mechloroethamine, streptozocin, phosphoamide, chlorambucil. Then it's not microtubule inhibitor, we already read. I can say among all microtubule inhibitor is most important because we already read, right? In this cytoskeleton we read. Keep it in your mind: paclitaxel, vincristine, vinblastin. Simple. Now you people no need to worry about the steroidal hormones, and I want you people to remember majorly friends, try to remember rituximab and cetuximab. These two are the two powerful monoclonal antibodies which we use in the cancer treatment, and these are also not required. Whichever 4, 6, 8, just I have marked 10. I want you people to remember these 10 examples without fail, which are powerful anti-cancer drugs. Never ever forget this. Okay, so friends, let's solve some of the PYQs. Yes, everyone. Okay, I'll zoom it for you, by that you can see nicely. Yes friends, question number 172, which is a part C question. Question number question number 172. Which one of the following combination is the correct pairing of ligands with their receptors? FGF, hedgehog, Wnt. We discussed everything friends. You can give the answers, no poll. Anyone? Yes, please. Yes. Why this much time you people are thinking? It's a very simple and direct question, no friends. Anyone? Anyone can give the answer? Okay, no problem at all. So friends, we have discussed, right? There are many growth factors are there. Very good, Saima. Very good. Epidermal growth factor, fibroblast growth factor, I have told you very clearly. Insulin growth factor, I have told you very clearly. Nerve growth factor. We discussed PDG, of platelet derived growth factors. Correct, na? Correct. So FG, these are all growth factors are going to utilize what kind of receptors, friends? We all know that it is going to utilize receptor tyrosine kinases or RTK receptor. I have explained it to you very clearly. Hedgehog, it is associated with the patched receptor. It's a very direct question, friend. Correct, Kavita. Wnt is the frizzled receptor. Hence the correct answer will be A, which is a part C. Okay. Then the question number 173. This is also part C. They are asking which is not correct, my friends. It's very simple question. Use the common sense. Don't be hurry, okay? It's a very simple. Right or wrong, try to understand it. It's not logical at all. They're asking which is not correct. You will get confused with the C. I am just giving hint to all of you friends. Option C is absolutely true. Teratocarcinomas prove tumors arises from stem cells without further mutation. The statement is true. I am just giving you hint. Try it. Even though it's wrong, please say it. Yes, anyone? Yes, Saima, Savita, Kavita, Padmavati, anyone? Shrikant. D. Okay, stemness genes can often function as an oncogenes. That statement is true. Okay, no problem. Okay, friends, listen to me. Cancer is often believed to arise from a stem cells rather than fully differentiated cell. My friends, so you only try to understand from the fully differentiated cell, we never get the tumor at all. Because friends, everyone please listen the statements carefully. The tumorous cell never undergo the differentiation. They said fully differentiated cell never cause tumor. Following are the certain views related to above statement. Which one of the following is not correct? Now first of all, everyone answer my question. First of all, see, this is a very simple question. Tell me, Saima, very clearly, stem cells will divide or not? First of all, stem cells will divide or not? Of course friends, stem cells will show the rapid division. Yes or no friends? Of course stem cells are going to show the rapid division, there is no doubt about it. But you only look into the option A, very simple, right? They said that stem cells do not divide. Absolutely wrong statement. It's a very simple question friends. Stem cells always divide. It do not require any changes to become a cancer cell. When, see friends, you have to understand one logic, right? Please listen the logic whatever I am saying now. If a cell is not divided, how it can give rise to a cancer cell? You only tell me. The cell has to be divided, but the cell has to be divided excessively. Then it can give rise to the cancer. What they said, stem cell do not divide, so they require a fewer changes to become cancer. No. You cannot create a cancer cell. You are getting my point. Here, in terms of this statement, here stem cells divide because of their excessive division or because of their excessive proliferation, they converted into cancer. So option A is wrong. The correct answer is A. Cancer stem cells can self-renew. Of course true. In the day before yesterday class also I told you, a self-renewing stem cell means what? It can give rise to itself. And same example, I don't know how you people neglected this option. And we discussed that it is going to give rise to a non-daughter stem cell. I know you people got confused here. What is the confusion you got? You got a confusion that cancer stem cells can self-renew. That is true point. Means this is a stem cell which is already infected with the cancer. It can undergo the irregulated division to produce itself. But whenever it is going to give rise to the other cell, the other cells are not. Other cells are what? Not affected. Why? Because everyone listen, they are non-stem cell. They are not the stem cells. Friends, it's a very small logic present here. In the first point, you can see cancer stem cell can renew. At the same time, see everyone listen, when the cancer stem cell got a self-renew, once again it is going to give rise to a stem cell only. There is no doubt about it. But whenever it is giving another cell, that is what? Non-stem cell. Non-stem cell never ever converted into a cancer. That's an important statement. This is right. As I told you, teratocarcinomas prove tumors. That's right. Stemness genes can often function as a oncogene. That's also true. Hence the option which is not correct is the option A. Please remember it. No problem. Yes. Question number 127. I will read it for you. I don't know why it did not come correctly. A tumor suppressor protein. Option A. It is the one whose function bring about regression of a tumor. Option B, friends. One where mutations are shown to cause or are associated with tumors. Option C. It is inactivated by the oncogenes. Option D. It inhibits the progression of the cell cycle by phosphorylating the cyclins. They just asked what is a tumor suppressor gene. Very direct question, friends. A tumor suppressor genes. Try it. You can tell a direct statement here. The question asked is what is tumor suppressor gene? I mean to say, what is the function they are asking? What's the function? Anyone. Right or wrong, no problem. Please say the answer. Kavita said A. Okay, Saima said B. Anyone else? Okay, very good. So the answer will be, friends, B. A tumor suppressor, friends, we read right. What is the function of a tumor suppressor gene? Tumor suppressor gene, it is responsible for what? Controlling the tumors. Correct na? It is controlling the tumors. I have already told you. Suppose example, if the tumor suppressor gene, take an example of a P53 we discussed. If the P53 got mutated, do you think tumor formation can be controlled? It cannot be controlled. You look into the option A. It is the one whose function brings about regression of a tumor. See, I can tell you one very important point here. I will agree with you. Option A and option B are most appropriate statements. Both are very close. But why the option B is the correct answer? Because, friends, you have to understand, tumor suppressor gene is the protein. If it undergo the mutation, this is the one which is going to recognize that mutations, and it will try to stop that mutation. It will try to stop the tumors which are associated with that mutation. I am requesting all of you once again, try to understand the statement. Why option B is the most important statement is an example of any tumor suppressor gene where mutations are shown. Wherever the mutations are there, it will recognize that mutation and it will associated with the tumors associated with that mutation, it will suppress that. So that's why this is the most appropriate. Okay, this is also right, there is no doubt about it, but it is not most appropriate, and it never been activated by oncogene because there is no role at all. It inhibits the progression of the cell cycle by phosphorylating cyclin. No, when the cyclins get phosphorylated, they get activated. First of all, this statement is wrong. Hence the correct statement will be, friends, option B. See, don't confuse, friends. Option A is correct, I am saying, no doubt about it, but the most appropriate is the B. Okay. It's a direct question, friends. Cancer causing genes can be functionally classified into majorly 3 types. Genes that induce cellular proliferation, tumor suppressor genes, genes that regulate apoptotic pathway. In the yesterday class, I have shown you a table also. Epstein-Barr virus that causes the cancer by modulating apoptotic pathway contain a gene having a sequence of homology with which of the following genes? That's the question. See, what they are saying is, I hope you are understanding. First answer. Try it. This is also part C question. Right or wrong? Say it. Savita, what's the answer? Saima, Shrikant, Kavita, Aishwarya, what's the answer? Padmavati, what's the answer? Oh, sorry. It's okay, friends. Right or wrong, say to me. Please answer, everyone. B. Okay, Saima said B. Anyone else? D. No. Okay, I want every one of you look into my question very carefully. See friends, please first of all listen and ask me if you really did not understand. Okay, please pay attention towards this. They are asking a question, Epstein-Barr virus, it is the one which can cause the cancer. It can cause the cancer. How it can cause the cancer? By modulating the apoptotic pathway. See, you only tell me by modulating the apoptotic pathway. So you have to understand which genes are involved in the apoptotic modification or a modulation. You know, right? Caspases are the one which stimulate the apoptosis. Which stimulate the apoptosis. It cannot be an answer. And you know very well, right? P53 is a powerful tumor suppressor genes which are required to control the cell division, to control the DNA repair, and if not, it will stimulate, it will always stimulate the apoptotic pathway. Same with the Bax protein also. I want every one of you remember originally what the BCL2 will do. Everyone listen. BCL2 also responsible for stimulating the apoptosis. Stimulating the apoptosis. But you have to understand the Epstein-Barr virus look like this BCL2. Now what BCL2 will do? It will allow the cell to undergo apoptosis because the cell got damaged. But what the Epstein-Barr virus will do? It looks like a BCL2. Instead of stimulating the apoptosis, it will inhibit the apoptosis. Listen to me. It will inhibit the apoptosis. Whenever it inhibits the apoptosis, the tumor cell never die. The tumor cell never undergo the apoptosis. Friends, very simple. BCL2 originally required to stimulate the apoptosis. This Epstein-Barr virus, so BCL2 allow the cell to die if it got damaged. But Epstein-Barr virus, it looks like BCL2. Instead of stimulating the apoptosis, it modulates the apoptosis. As a result, what is going to happen? The cell never undergo the apoptosis. It is converted into a tumor. Friends, everyone understand this question or not? The answer is BCL2. Clear everyone? Please say yes or no. Okay. Friends, I hope it's clear for every one of you. Okay. So what I'll do is I will take the antibiotics class tomorrow itself. Why? Because in half an hour I can't finish it. Okay. I'll take tomorrow full-fledged class at sharp 7:30. 7:30 to 8:30 we will finish the antibodies and we'll solve the PYQs related to the antibodies in the immunology concept. Okay, I hope the discussion is useful, friends. I'll see you tomorrow sharp 7:30. Once again, everyone try to revise the antibodies. We will solve many kind of PYQs which were asked from this particular antibodies. Okay. Okay, friends, please revise antibodies without fail tomorrow. Okay, I'll see you in the tomorrow class. Thank you all. Thank you so much. Thank you. BCL2 is an anti-apoptotic. You cannot use a word anti-apoptotic. No, you can say it here. Yeah, you can say anti-apoptosis. That is true statement. That is true. True. Correct here. It's an anti-apoptotic. It is modulated its action. Who? Epstein-Barr virus. Because of modulation of its action, instead of controlling the apoptosis, it loses the ability of controlling the apoptosis. You're getting my point? Yes, that's true. Okay, friends, I'll see you in the tomorrow class. Thank you all. Thank you. Please attend tomorrow sharp 7:30 p.m. We will continue with the antibodies only. Okay, we'll finish it. Thank you.